Professor Snap, where we aim to make A-level biology as simple as a cell. So today we're looking at the ultrastructure of eukaryotic cells, including nucleus, nucleolus, ribosomes, rough and smooth endoplasmic reticulum, mitochondria, centrioles, lysosomes, and Golgi apparatus. We'll also be looking at the role of the rough endoplasmic reticulum and the Golgi apparatus in protein transport within cells, including their role in the formation of extracellular enzymes. Cells can be described as either prokaryotic or eukaryotic cells. Eukaryotic cells have a more complex ultrastructure. Multicellular organisms such as animals, plants, and fungi usually have eukaryotic cells. The cytoplasm of eukaryotic cells contain membrane-bound compartments called organelles. What makes eukaryotic cells distinct from prokaryotic cells is the membrane-bound organelles and a nucleus, which are features that prokaryotic cells do not have. And also there's a distinction in the ribosomes as well. So eukaryotic cells have an 80S ribosomes, and prokaryotic cells usually have 70S ribosomes. However, there are differences between animal cells and plant cells. So both animal and plant cells, they have eukaryotic cells, but even then they have some differences in their structure. So plant cells have a cellulose cell wall, they have large permanent vacuoles and chloroplasts. However, animal cells contain centrioles and some have microvilli depending on the function of the cell. So you would not see animal cells with a cell wall, permanent vacuoles, or chloroplasts. Those are features in plant cells only, and some animal cell specific features are centrioles and microvilli if it has a specific role in the body. Focusing on the cell surface membrane, also called the plasma membrane. So the cell surface membrane controls the exchange of materials between the internal cell environment and the external environment. The cell membrane is partially permeable and is made of a phospholipid bilayer. Focusing on the nucleus. So nucleus is present in all eukaryotic cells and they also have a double membrane. And this membrane is called the nuclear envelope. The nuclear membrane has nuclear pores and these allow mRNA and ribosomes to leave the nucleus and enzymes to enter the nucleus. And from topic 2, which is now fully uploaded, you would know that it is important for mRNA and ribosomes to leave the nucleus for DNA translation. The nucleus contains chromatin, and chromatin is the material from which chromosomes are made. And just to remind you guys, chromosomes are made of sections of linear DNA wound around proteins called histones. The nucleus has an internal spherical structure called the nucleolus. And if you see like past paper questions and you see the nucleus, the darker circle that you will see is the nucleolus. So do keep an eye out for that if you're asked to like mark specific structures in your question. So yes, these will appear darker than the nucleus under a microscope. The nucleus is the site for ribosome production. Focusing on the mitochondria, which, as we all know, is the powerhouse of the cell, but you cannot write that in A-level biology because you're not going to get any marks. But yes, mitochondria is the site of aerobic respiration. Mitochondria have a double membrane, and the inner membrane forms folds to form structures called cristae. And the matrix of mitochondria contains enzymes needed for aerobic respiration. The mitochondria also contain small circular pieces of DNA and ribosomes as they are needed for mitochondrial replication during cell division. Looking at ribosomes. So ribosomes are found free floating in the cytoplasm or found on the rough endoplasmic reticulum, and they are the site of DNA translation. And each ribosome is a complex of ribosomal RNA and proteins. In eukaryotic, so this is a distinction that you should know. So in eukaryotic cells, you'll find 80S ribosomes. And in prokaryotes, mitochondria and chloroplasts, you will find 70S ribosomes. So these are like two different sizes for ribosomes, if you want to know what it means. But you don't have to know any more detail about the size. So now we're looking at the endoplasmic reticulum. So the endoplasmic reticulum can either be rough or smooth. So one is the raphia and the other one is the smoothia. So raphia is covered in ribosomes and are formed from folds of membrane continuous with the nuclear envelope. The aria processes proteins made on the ribosomes, and the smoothia is also formed from membrane folds, but its function focuses on the production, processing, and storage of lipids, carbohydrates, and steroids. And you should know the difference between the raphia and the smoothia because there are so many past paper questions on them. And the smoothia is smooth, just like the name says, as they do not have ribosomes on its surface. So, so if you look at any like questions where they ask you to label different features on the cell, and if you're confused between the raphia and the smoothia, you need to look for the one that does not have a smooth surface. So if it does not have a smooth surface and it has dots on them, it's most likely to be the raphia. And if it's smooth with no dots, it's a smooth ear. And just to remind you guys, the dots on the raphia, that's ribosomes, but you don't have to label them specifically unless they ask you to. So now the Golgi apparatus, which you guys should also definitely know. Once again, so many past paper questions. They usually come in 3 or 4 markers. The Golgi apparatus is made of flattened sacs of membrane, and if you guys look at like labeling questions on past papers, it's usually in the shape of a Wi-Fi symbol, and it's also smooth like the SCR. So it's smooth like the SCR, but the difference is it's more like a Wi-Fi symbol than a random structure. The Golgi apparatus modifies proteins and lipids and packages them into Golgi vesicles. The vesicles then transport the proteins and lipids by attaching and fusing with the cell membrane. So now, looking at the function of raphia and Golgi apparatus. So organelles are specialized parts of a cell that carry out a particular function in the cell. Nucleus, rough endoplasmic reticulum, Golgi apparatus, cell surface membrane, and ribosomes are organelles involved in protein synthesis. And you guys would know this from topic 2. And if you want to revise, do check it out. It's been uploaded. So transcription of DNA happens in the nucleus and translation of mRNA happens in the ribosomes, which is outside the nucleus in the cytoplasm. So the proteins produced by ribosomes on the raphia are passed through the rough endoplasmic reticulum where they are folded and processed. Are processed in the raphia and then they are passed through the Golgi apparatus in vesicles, which fuse with the Golgi apparatus and releases the proteins into the Golgi apparatus. So they are transported in vesicles. And then the proteins are modified and are prepared for secretion by packaging and leaving the cell or transport to another part of the cell in vesicles. So they enter the Golgi apparatus in vesicles, they get modified and prepared as needed, and then they leave the Golgi apparatus to wherever it needs to go in vesicles as well. Looking at lysosome, which is one of those parts that people often forget to revise, but again, it's quite safe to learn about it. So lysosome is a type of vesicle that contains hydrolytic enzymes. The lysosome breaks down waste materials in the cell, and the function is more dominant in the cells involved in immune systems. So these can come up as 1 markers where they ask you what's the function of a lysosome, or they give you a function and asks you what organelle in the cell that performs this function. So it's quite a good one to know as well. So now we're looking at centrioles. The centrioles are made of microtubules, which are hollow fibers. Microtubules are filaments of protein that move substances in the cell around and supports the shape of the cell. When 2 centrioles are at right angles to each other, you get a centrosome. And the centrosome organizes the spindle fibers during cell division. Centrioles are found in animal cells, and disclaimer, they are not found in plants and neither are they found in fungi. So that is all from Professor Snap today. Hope you found this topic useful. If you have any questions, please let me know in the comments below and I will get back to them as soon as possible. And don't forget to like, share and subscribe.